So I just first want to say thanks for letting me speak and present my work. The title of my talk today is Testing SARS-CoV-2 in Wastewater as a population level COVID-19 surveillance tool to complement clinical testing in Metro Vancouver. And so I just want to highlight in the beginning, my work was primarily focused on research and development and validation of methods to detect enteric pathogens in wastewater. And the reason why we really like enteric viruses is because one, they infect the GI tract and they cause gastroenteritis. And so therefore, they end up, you shed it through your stool, they end up in the wastewater and are easy to monitor because they have a low infectious dose and they have high viral shedding in vomitus and stool, but they survive for a really long time in the environment. They're really hardy viruses. So they're very useful viruses for us to actually study and try to connect the environment to the clinical outbreaks. So the viral dispersion and rate and fate of the receiving environment of the viruses after treatment in wastewater is unknown. So we were developing methods to detect these viruses once they enter the treatment plant and then before they're released into receiving waters like beaches and rivers to kind of get a view of the risk for public health or if they would end up in, say, if in the rivers or irrigating crops, what is the risk there? So I had a pipeline already set up because we had a long existing collaboration with Metro Vancouver. So when—
Sorry, Melissa, sorry to interrupt. When you advance your slide, please let me know.
Yes, can you advance?
So which slide are you at now?
3.
Okay. Yep. Thank you. Sorry.
Yeah, that's okay. Did you were you able to show the one before?
Very briefly.
Okay. Okay. All right. So I just want to mention here that wastewater surveillance is a vital tool. This is not a new concept. Some previous examples is in Israel, they test for a polio virus on a routine basis. And in our own backyard, we monitor for illicit drug use. This is done by the Public Health Agency of Canada. And we also monitor for enteric viruses. In other countries in Europe, this is a tool that's more widely used. So it really wasn't until COVID that this tool became recognized as a valuable tool that can be used around the globe. So the picture on the left kind of shows you like all the different communities that are involved in a catchment population. And what I mean by that is that all the waste goes into a system and it all ends up at a wastewater treatment plant that is specific to that community. So you have houses, you have, your downtown buildings, you have community-wide facilities like shopping malls, hotels, events, offices, industry, and then specifically you have some community services like hospitals, universities, and prisons where you can do building scale surveillance. So all this ends up into a wastewater treatment plant, and you can monitor from collecting from the wastewater treatment plant, you can monitor the presence and dynamics of a pathogen in communities. And this data can provide early detection of disease prior to an outbreak and includes individuals with symptomatic and asymptomatic infections. It can capture the emergence of new strains or variants, and it's the beauty of it is it's independent of clinical testing. So access to clinical testing, and when clinical testing gets reduced, like in our current state. Next slide. And so on March 11th, which seems like a really long time ago, what happened with the pandemic is that we basically all of our research was put on hold and all hands were on deck to study COVID, especially the BCCDC. We are public health. And so our only focus was COVID. And so essentially we had to switch gears. And so as many of you know, SARS-CoV-2 RNA, it's detected in the feces. This COVID was at first recognized as more of a respiratory illness, but now we kind of see it as much more affecting multiple, it's more systemic. But a significant portion of COVID-19 patients display gastrointestinal symptoms, and this can be vomiting or diarrhea. And so this is old data. So at the time, 67% of infected patients with the first strain shed SARS-CoV-2 in their stool. Symptomatic and asymptomatic shed. And there's also persistent shedding even after the virus is cleared from the respiratory tract. So in like an elderly patient, you shed SARS-CoV-2 in your feces for a prolonged period of time. I think there was one patient that shed for 90 days. And so this makes it very easy to detect in the wastewater. So we found it in the feces, we found it in the wastewater. And so then we knew that we could run a surveillance project. And so if you can advance to the next slide, we have 2 poll questions. Okay. So do I share the results? Or do you guys have them up?
Yeah, I can share the results.
Okay.
Yep. And then after we share this, then we can start the next poll. If then you can let me know when to start.
Okay. Or do you want to go over the first question first? Can I review the first question?
Yes, please do.
Okay. So there is no evidence of fecal oral transmission. So that let's go to the next question, and then I'll expand on this. Oh, it seems like the same question. Okay. All right, so I just want to highlight that for poll 1, I just want to say that there's been scant evidence. It depends on what you mean by proven infectious in stool. What people have done is there's 3 groups that were able to grow SARS-CoV-2 in like cell culture from a stool sample, but they weren't able to repeat the results, and the conditions weren't they weren't very representative of what is physiologically relevant or representative. And so nobody so far has been able to continue to grow SARS-CoV-2 in stool samples to a point where it is infectious and taking over cells at a high rate. I think with the cell culture, maybe it was a very small amount, and it wasn't something that could be repeated. When it comes to poll 2, so as far as fecal oral transmission, there's no evidence that there's you can get it from touching, say, a doorknob from a bathroom, and put in your mouth. If you put your hands in your mouth, you're not washing your hands, that you could get SARS-CoV-2 that way. Fecal oral transmission is usually from enteric viruses, and this is not an enteric virus. And so far, there's no evidence leading to fecal oral transmission. So we'll just continue on to the next slide. So I just want to point out, because this is a group in the Netherlands, these researchers were the first to report wastewater for COVID surveillance. Essentially, what they showed is that there was a correlation between SARS-CoV-2 detection in wastewater and COVID-19 case counts. What was really instrumental is that they were able to detect SARS-CoV-2 in wastewater 6 days prior to the first case reported. So it's a good early indicator if you were testing your wastewater frequently, like every day. And it's also very useful to monitor the transmission of COVID-19 in communities. So with that, soon researchers around the globe started testing SARS-CoV-2 in wastewater to use as a surveillance tool. And so this is a map provided by COVID Poops. It's a website that you can look and see all the different people around the world that are doing this work, and there is a lot. If you look at the states, every single state seems to have testing done, and the CDC has a national surveillance program. BioBot is huge, and everybody sends their samples to them as well. And in Canada, we're doing also pretty good as far as having at least I think almost every province has testing for wastewater for COVID surveillance. And Europe is also quite prominent, UK especially, and the Netherlands, and also Australia. And then so this is kind of just showing our own backyard. We have a lot of work happening in our own backyard as well. So it's a thing that was popular in Europe, and now it's been now being done across the globe, and it's well recognized by public health. So if you can advance to the next slide. And then to the research objectives slide. And so the research objectives in 2020, when the pandemic was declared, we wanted to develop the methods basically we're developing methods to detect and quantify SARS-CoV-2 in wastewater samples, leveraging the existing methods already had for enteric viruses. So there's a timeline from May is when we started until September. And what the outcome of that was method validation. I entered or participated in a few Canada-wide interlab studies to have the methods tested and validated, so that we can roll them out a surveillance program. And then quickly after validation, we started a proof of concept study to conduct a longitudinal prospective study to determine and quantify SARS-CoV-2 in wastewater samples from all 5 of the wastewater treatment plants. This was from October to December, and the outcome of this is that we demonstrated that it was ready for use as a complementary COVID-19 surveillance tool and for reporting to public health. So the methods that go into this is that you have the sample collection. So at the wastewater treatment plant, you have this 24-hour composite machine that is collecting the samples every hour for 24 hours, so you can actually get a day's worth of data. And we do this 3 times a week, and everything is kept at 4 degrees because SARS-CoV-2 likes to be cold. And then the next thing we do, because in Vancouver, we have a lot of sand and grit in our wastewater. So we spin everything down to get the large particles out. And then we take the supernatant and we process it to get a viral concentrate. And we do that for 2 reasons. One, we're trying to work with a large volume, which it used to be larger than 15 mils. It used to be like 60 liters. And then we got down to 1 liter and then 100 mils and now 15 mils. So we've definitely improved our methods. But the purpose is to get it down to microliters, because you need it down to microliters in order to work with the downstream molecular applications. And so we use this Amicon device, and it's based on pore size. So what it does is it selects the viruses, because anything that is smaller will go through. So you'll get like a viral concentrate. And then from the viral concentrate, you're extracting nucleic acids. And those are used to test SARS-CoV-2 via, we use the clinical assay for the BCCDC targeting the E gene. I just optimized it for wastewater and then added standard curves so that we could quantify the amounts and get the units of copies per liter. Next slide. So now we're at poll question 3. And I just want to ask that, there's a blank here. It's a question, and then you have to fill in the answer. If you could do that, on your own during this poll, that would be great. So essentially, the question is in regards to safety and handling and processing wastewater samples to obtain the viral concentrate. All procedures are performed in biosafety level what?
Marissa, the question is in the chat box. So people can, reply their answers in the chat box and see the question.
Okay. So I am not able to see the chat box when we're done, if you guys want to elaborate, and then or I can just answer with the question and explain.
Yeah, we are getting answers from the audience, a lot of 3, some 2. Any last minute answers? Yeah, majority 3.
Okay, so the answer is it's biosafety level 2 with unidirectional airflow and biosafety level 3 precautions. So we're not officially at a BSL 3, but we have to use the precautions. And so any procedures where you're concentrating virus, and you have to, take the sample out of the biosafety cabinet and then put into a centrifuge, you could possibly aerosolize virus. So what we do is our PPE that we wear is for level 3 precautions, and we wear a respirator mask. We also have to use, for our swinging bucket centrifuges, we have to use safety caps. And like there's safety cups and caps that are closed to reduce the risk of exposure to the laboratory. And then we also tape our gloves to our gowns. There's a whole bunch of safety that goes into it, just to make sure that you protect yourself just in case. So that is the answer to question 3. You can go to the next slide. This is the regional wastewater treatment plant system map. So each wastewater treatment catchment area is color coded. So on the top you have this orange zone, and it's called Lionsgate, which is for the North Shore. Iona is in red, and that's Vancouver. Then you have Lulu, which is basically just Richmond. And Annistis is our largest wastewater treatment plant. It treats over 1 million people, and it includes many different regions. So the dark green one is Northwest Langley, and it's our smallest plant. So it's very specific to the township Langley. So the beauty about this is that all 5 plants cover 50% of BC's population, and it's located within 2 separate health authorities. So Annistis would be considered Fraser Health. That includes Northwest Langley. So that's more like Fraser North and South. And then the others are Vancouver Coastal Health. And so this, if you want to advance to the next slide, this is a nice little cartoon that shows an overview of how COVID wastewater surveillance works. So first you have an infected individual. It's SARS-CoV-2 is shed in the stool. It ends up into the wastewater network. And then once we collect the samples, the virus is then tested for SARS-CoV-2, and then it goes to the epidemiologist for modeling, and then to the public health, for, reviewing the data and making decisions. And so the one thing that we have, a large benefit of working at the BCCDC is our connection to public health. So we have a whole team of epidemiologists, we have data analytic partners, and then we also are very well connected with the public health officials. So it was very easy for us to have the surveillance shared with public health quickly. And so essentially, if you want to go to the next slide, I'll just zoom in on what our team does at the BCCDC. And so essentially, as soon as the samples come to my lab, I have my technicians and students, they process them, they get the data, they go through all the calculations, and then they send me a summary, and then that summary is then sent to, it's reviewed by the BCCDC epidemiology and data analytic partners. And so all of the qPCR positive COVID-19 cases are mapped to the sewage catchment region. And so that's done by our medical geographer, Sunny Mack, who's very, very talented. And then Dr. David McVeigh, who is a public health physician and epidemiologist, him and his data analyst, Michael, they compare the SARS-CoV-2 wastewater data to the instance of community cases. And basically we have an automated pipeline that once each of these steps happens, a button gets pressed, and then data comes out, and then graphs are made, and then figures are made for my reporting. So the next thing that happens is that I take that PowerPoint that is automated, alter it, and then write the key messages of what's happening for that week's data. And then I disseminate that knowledge to the provincial health officer, to the medical health officers for each region, epidemiologists at the regional health authorities, and also to our stakeholders and internal staff at the BCCDC for the public health lab. So I just want to show you some results. I am purposely showing, it's 2021 data, a time series, but I'm purposely showing you data that is cut off before December. And the reason for that is because with Omicron, we had a huge spike, and it was so large that we if you graph the data together, you kind of lose the visibility of the trends that were taking place before Omicron and after. So I just want to demonstrate here. This is for Fraser Health. You have Annacis Island on the top, Northwest Langley on the bottom. And so the axes on the left are for the daily case counts of COVID. And also on the right is a viral signal. It's the SARS-CoV-2 concentration. So the dots are for wastewater, the blue line are for cases. And another reason why I'm showing this is because of the correlation values. Our data correlated nicely with the case data for 2 years. And then the Omicron peak kind of went, things went a little weird. So I just wanted to show you how well this was working before Omicron. And so essentially what I want to say is that each wastewater treatment plant is different. Annacis, there's a nice correlation between, the trends in cases with the wastewater. And this is, a peak that was happening in, mid-January all the way to March. Then things died down, and then things went back up with Delta. And so what you can see here is that we did have pretty good correlations, except for this Northwest Langley. We have a bit of the day's a little bit messy, and it's we're still trying to figure out the reasons why. The data is not always going to be perfect, and that's why it's good to know how to, your reasons for some of your noise, and also, which I'll talk about later, normalizing the data. And so next slide. We have Vancouver Coastal Health, Iona on the top, Lulu on the bottom. Pretty good correlation between cases and wastewater data over time. Same trends for our, our peaks. And the last one, if you want to go to the next slide, shows Lionsgate wastewater treatment plant. And so overall, I just want to say that the SARS-CoV-2 signals in wastewater are consistent with the number of case counts. And so the feedback I heard from the medical health officers that this data is really reassuring, because it's independent of clinical seeking behaviors. And it further shows that when restrictions were advised, case counts and wastewater signals dropped at the same time. And the same could be said for low case counts and wastewater signals during the times when we had good vaccine coverage. So what we are looking for is a discrepancy in these two, when the data is not, when it's not going in correlating in the same direction. So that's when, if we get a spike that is early, from the case counts, then that is our early indicator for a future outbreak. And that's when we report to the medical health officers immediately. Next slide. And so now we're going to get into a can of worms that, once we open this up, there's a lot that can be discussed, and a lot of, there's, limitations, and there's also, benefits to this. So the old school way of doing things is normalizing your data to daily wastewater flow. And so essentially that adjusted data for, to account for relative volumes that are going into the wastewater treatment plant. So that's going to account for any sort of rainfall events, any heavy rainfall events, or some snow melt, or say you have a spill and that goes into the plant, and it's going to tell you if something isn't functioning normally. And then we have what these molecular markers, they're fecal markers. They're something that's very trendy in the recreational water field, and also with enteric viruses. Actually, this pepper virus was, was originally going to be used for enteric viruses, and enteric viruses are replicated in the gut and shed solely through the feces. And so it seems like a very appropriate marker for enteric viruses. It's very interesting because essentially you ingest this virus, which is not harmful to humans, by the way, in large quantity by consuming processed pepper products. And so subsequently, it will pass through the human digestive system and at wastewater. And what we find in Vancouver, talking with the Public Health Agency in Canada, Vancouver has like the highest amount of pepper virus in wastewater. Like we eat a lot of pepper products. So it's a very high signal for us, which is hard to use for normalization. We do consume it quite a bit. When you're consuming the pepper virus, it could also be dependent on diet. So that's one thing that is a limitation. It was supposed to be seasonally stable, and what we find in Vancouver, because we do have such high rainfall, that it's not as seasonable, or like it's not seasonally stable. It gets diluted with rainfall, and so we're still trying to figure out how best to use this data. So the data looks different per wastewater treatment plant and per province. So we each have to decide the best way to normalize our data. And so that is still something that is currently being discussed. And hopefully we can resolve that quite soon. Next slide. So these data, this is Fraser Health. This is normalized to flow. So this is data showing, the Omicron wave, which you can see that gigantic peak in December, and then it going back down. And then currently the last dots are what we're actually from last week. And so for Anasis, we are seeing a little bit of an uptick in our values. We are currently adjusting our graph so that we can see the trends in the data before and after the Omicron. I think we're just going to shave off the top peak of this Omicron wave so that we can see these values at a better trend. We zoom in on them for reporting. But for today's purposes, these are the graphs I have. And so essentially, Northwest Langley looks still a bit, it looks pretty stable comparative to past weeks. For Vancouver Coastal Health, all 3 wastewater treatment plants, it does look like the values are increasing. It's a slight uptick, but I don't know if you guys have been following the news. We want to watch these data closely because it seems like the values in wastewater are increasing for places like Ontario and Alberta. And so we're just looking at this data closely to see if this increase becomes a trend. So did you advance to the Vancouver Coastal Health slide?
Yes, I did.
Okay, next slide, please. All right, so now because of time, I'm actually going to just tease you because I don't have time to talk about these exciting projects, but hopefully you can invite me back to talk about building scale monitoring. We are monitoring wastewater from 5 buildings on UBC campus. We collect samples 3 times per week, and we do also I report to the medical health officers and the UBC stakeholders on a regular basis. So a majority of these sites are dormitories, and this data is actually very very useful because it is targeting a building. And so I just want to show you because this part is exciting to me. These are the little sewer cages that we chuck these down a manhole. Inside it, that little white thing is a membrane. It's electronegative membrane. They were basically printed by a 3D printer. They're cheap, and they were developed by our colleagues at Dalhousie. And essentially that electronegative membrane, it's negative, so the viruses will stick to it. So basically what we do is we put it onto a wire, and then we chuck it down a manhole, and then we let it, if you want to go to the next slide, it'll show a great picture. We let it sit in that manifold for 24 hours. It collects the sewage from that building. Next day, we have a team that helps us take it out, and then we take it to the lab, and then we test it. So it's a very cool device, because some of the old devices that we had for passive sampling were like using tampons and weird cheesecloth things. So I really like the sewer cage. Well done, Dalhousie, Emily, to be specific. And so if you want to go to the next slide, this is the next thing I'm going to tease you with, is that what about testing for wastewater samples for variants of concern? We have a project with Dr. Ryan Stills in UBC Civic Engineering, and his PhD student, Shuan, has been doing this for all of our positive samples, and it's very exciting data that is complementary. So perhaps we can present that another time. And then quickly, I'm just going to quickly go over the summary. So for surveillance, first you have to validate your methods if you want public health to trust and rely on it and use it. Our surveillance ended up being for 5 wastewater treatment plants in Metro Vancouver, which nearly covers 50% of BC's population. Been doing this since 2020. And the significance is that it monitors the presence and trends in the incidence of community cases. It includes individuals with symptomatic or asymptomatic infections, and it's independent of clinical testing. And the other thing is that there's a key knowledge translation and transfer component. We report the data, weekly to public health officials, our stakeholders, and we also make it available online. So I ensure that there's, the knowledge is transferred for the public health to be able to grasp it and use it. And then Metro Vancouver has made this really cool dashboard that's interactive, where you can hover over each of the regions. So if you live in Richmond, you hover over Richmond and you see what's happening with COVID in your community. So that was a very nice little add-on. And then so last, advance to the next slide. It's wastewater surveillance is a vital tool. I just want to highlight that this provides a community level surveillance data that complements traditional surveillance programs. It's also an approach that's independent of healthcare seeking behaviors. And so now it's the best surveillance data that we have, given that the current limitations that have been put into place for clinical testing. And this tool is highly adaptable to be leveraged for a rapid assessment of emerging threats and preparedness for pandemics. And so my last slide is really just saying what we're doing beyond COVID. So we're leveraging this to basically test other pathogens, like go back to Enteric. Norovirus is a big one. Respiratory pathogens like influenza and RSV. And also another big interest is antimicrobial resistant organisms. And the other thing you can do with genomic sequencing is that you can look for the unknowns. So existing and emerging pathogens, so that you can be prepared for what's to come. It is kind of a needle in a haystack approach, but is useful for planning for treatments. And then also we're doing the targeted approach, so building scale surveillance and also ad hoc sampling for outbreak management. And then with that, I'll end. These are all of my acknowledgements. I'll leave them up for a bit because everyone at the BCCDC, my boss, my mentor, Dr. Natalie Price told Jackie, and then everybody else that is involved in helping, because we're rolling some to routine services in the environmental microbiology lab. So that's been a key component. All the epidemiologists and data analytic partners, Metro Vancouver for providing the samples, and obviously UBC for our sequencing of variants project. And last, this is just, I'm putting up my contact here. If anyone wants to ask me questions, feel free to do so.